Welcome to General Biology 1. I'm Joyson Hernandez. For today's topic is the cell cycle interface, mitosis, and cytokinesis. First up is the interface.
GAP1 or G1 phase or first growth. This phase is the cell growth and building blocks for DNA replication. Aside from the cells to grow or to double its original size, it also produces a lot of proteins, energies, and organelles that will be needed in DNA replication later in the next phase. This phase, typically the longest phase in the interphase that can last around 9 to 12 hours, and around 30 to 40% of cell cycle, this time is spent in this phase. Next is the S phase.
S phase are known for synthesis. This phase is responsible for the synthesis or replication of a complete copy of DNA in its nucleus. It only begins after the G1 phase. It doubles the genetic materials before it enters the M phase and allowing that there is DNA to be split into daughter cells. This phase is second to the longest phase and takes up only around 6 to 8 hours to complete. Next phase is the last phase, which is the G2 phase.
GAP2 or G2 phase or second growth. This is the last stage and it is all about the preparation and inspection of the cells for the M phase. The cells need to grow some more, provide molecules that needs to divide, assembly of the microtubules that will make up all the important structures called the spindle, and duplicate its organelles that each daughter cells get everything it needs. This phase only takes around 3 to 4 hours to complete. Interphase is complete. Next is the mitosis or the M phase.
The first phase of M phase or mitosis, the prophase. This stage where the nucleus disappears, spindle apparatus form, and DNA condenses into the chromosomes. The chromosome condenses until it becomes shorter and thicker that makes them to move easier in the cell. The nucleus dissolves and the centromes begin to move with the chromosomes, or forming in the middle of the cell by the mitotic spindle. In this stage, the chromosome consists of two identical parts called sister chromatids and stay connected at the centromere.
Metaphase. The stage is where the chromosomes align in the center of the cell. The chromosomes are captured and lined up by the spindle in the middle of the cell that can be called as metaphase plate. The two kinetochores of each chromosomes should be attached to the microtubules from opposite spindle poles. Each sister chromatids are attached to the spindle fibers originating from the opposite spindle pole and waiting for some signal for the next stage.
Anaphase. This stage is where the separation of breakdown of sister chromatids begins. The sister chromatids, making up each chromosome, are pulled apart and begin to move away from each other under the control of the spindle fibers to the opposite poles and form a bundle where the centriole located. Once they are pulled apart, the chromatids are now considered individual chromosomes. When the anaphase this stage is complete, the last stage begins.
Telophase. This stage is where the chromosomes already placed in the opposite poles. Recondensation begins and mitotic spindles break down. And the mitotic spindles are to decondense and return to the stringy form. They loosen up and spread around the nucleus because they no longer will need to move around. Nuclear envelope materials will form around each bundle of chromosomes. The cell cycle.
That's all for today. I'm Joyce Ann Hernandez.